Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Habib. I am here with JP. How are you, sir?
Doing okay. Hanging in there.
Yeah, we missed you on our last recording, and we're excited to have you sort of back and excited to jump into a topic on longevity, but a specific gene that recently has come into the research world and become a very important spot in promoting longevity overall. I would love for us to dig into this, but before we do, just defining kind of what we're talking about here is the genetic predisposition towards longevity or long life, right? So it's this specific gene called the FoxO3 gene that has been identified as a particular gene that drives extremely long length of life and
healthy life.
So let's talk a little bit about this. Let's start off with the genetic predisposition to living a long life overall, and then let's dig into the FoxO3 gene overall.
Sure. So there are, as many people know, there are different alleles when you have a gene. And so what is an allele? An allele is a slightly different version of that gene. So for example, blue eyes and brown eyes, those are two different alleles of the same gene. Now, in the case of blue eyes, you're talking about a gene that's malfunctioning, so it's not actually producing anything. But in many cases, you have two different genes that are slightly different. They may have a point mutation, in which case one nucleotide is different, or in some way they've switched an amino acid. Maybe it's more than one base pair that got switched, but it's just different, but it still serves the same purpose. And it just does it in a slightly either less efficient way or a more efficient way. Now, in the case of FoxO3, which we'll get into what that gene does and what that protein is useful for, and it's a lot of things, that gene can come in different flavors, sort of like ice cream, comes in different flavors. And there are some of those that are associated with, as you were describing, a longer, healthier life. So, we'll maybe attach in the show notes what that gene specifically is, or that allele is. So if you want to get tested, you can find out if you have it or not, and you can celebrate if you do. You can have yourself a little party. But at the end of the day, FoxO3 is what's referred to as a nuclear transcription factor. So, and it's been around forever. I mean, it's ancient. Flies and worms have this. And what it does is it is responsible. It's deeply embedded in how cells respond to stress. And as a result, how well it deals with challenges. So think of it as a master regulator that's sort of integrating various different signals that include mitochondrial oxidative stress states, growth pathways, mTOR that we've talked about before, nutrient sensing, and making sure that all of the right genes are being expressed so that you can survive some stress.
So it's a stress response specific gene, and based on the flavor of that gene, you're going to have some level of vulnerability or tolerance to specific stressors that are occurring. Is there any difference in the type of stress that FoxO3 is going to respond to specifically? Is like the type of stress going to have any role to play, or is it just overall how your body is responding to the stressor that drives it?
The answer is a qualified yes. In general, though, those stresses will activate FoxO3. Now, FoxO3, you got the gene, and that gene's coding for a protein.
Yes.
That protein, the word Fox, comes from this fork head structure. So if you think about it, sort of take your two fingers and make a peace sign, and that is the fork head structure that attaches to DNA. So what the gene is coding for is a protein that moves into the nucleus and out of the nucleus, depending on whether you're in a stress state. And the way the protein knows whether to be in one place or another is by whether or not it's been phosphorylated in certain places. So whether or not you tag a phosphate group onto it in certain amino acids. But basically that protein slides into the nucleus, binds to the DNA, and then facilitates or enhances promoter structures and other proteins that will bind to that DNA and leave it exposed so that it can be copied. Now, if it's not there, then it's less likely that that gene's going to be copied. And the genes that it's promoting, so this FoxO3, protein is now promoting all of the stress response, positive stress response genes that need to be transcribed and translated in states where there's, stress.
Okay, so that makes sense. Now we have this gene that's coded for the protein. That protein is determinant whether it's going to be in the nucleus or in the cytoplasm and moves in and out based on phosphorylation sites being activated, and then plugs in and turns on these genes that are coding for stress response tools, things like cortisol, I imagine being one of them, particularly adrenal gland specifically, right? Likely driving a little bit more of the glucose activation, a little bit more in certain areas. I imagine there's going to be some effect in the liver, some effect in the pancreas, slightly different genes being activated in different tissues, but essentially promoting the ability to handle a stressor in the overall holistic body. Make sense?
Yes, and what you've described in the liver and in circulation are things that are happening outside the cell, and those are really important signals for FoxO3 and its activity. But it's also activated by things that happen within the cell, and one of the really big ones is something we've talked about before, which is AMP kinase. So AMP kinase is an enzyme that is activated when there's not enough nutrients in the cell. So the cell feels starved. It's probably a little aggressive to call it starved. Let's say in a fasted state. What happens is you've got ATP, and ATP is constantly being generated when you have nutrients and energy sources present in the cell. And the mitochondria are churning out those ATP to be used as little battery packs. And the battery pack, when it gets quote unquote used up, it moves from an ATP, which is adenosine triphosphate. So it's got 3 phosphate groups attached to that nucleotide base. And then when that one phosphate at the end gets transferred onto something else to give it energy, now you've got ADP, adenosine diphosphate. Now that is what's getting recycled into making more ATP when you're in the mitochondria or in the cytoplasm. You can do it through.
That's in general, right? Not when we're in the fasted state, but rather in
Right.
general, when we have nutrients, we're going ATP, ADP, and back to ATP more easily. But AMP kinase is going to work differently because the stress level is different.
Exactly. So when you don't have enough energy and the mitochondria are sort of struggling to make the ATP, there is another mechanism by which your cells can create ATP by taking 2 ADPs. Now, if you think about it, an ADP is the adenosine base plus 2 phosphate groups. Now, if you have 2 of them, together they have 4 phosphate groups. What can happen is you can transfer one of the phosphate groups from one of the ADPs onto the other ADP and you get an ATP. So it's an alternative way of making an ATP, but it takes up 2 ADPs to do that. And what's left behind is AMP. Yes, and that's from AMP kinase. What happens is AMP kinase is this enzyme that's around measuring, sensing how much AMP is around. Now, there shouldn't be very much if you've got lots of nutrients and you're functioning at a high level of energy production. But when you're at that lower energy production rate because of being in a fasted state or being without nutrients, that AMP starts to build up. And the AMP kinase then activates and it says, hey, what we need to do right now is we need to break down all the garbage, all the debris that's been left in this cell. We call that autophagy. So AMP kinase is recognizing a stressed state. And by recognizing that stressed state, AMP kinase actually, as the enzyme, is actually phosphorylating. So it's using one of those ATPs. It's phosphorylating the FoxO3 protein at specifically serine 413. And that then promotes the FoxO3 going into the nucleus and turns on those survival mode, genes. And that will promote catabolism and autophagy. It's actually promoting the production of all of the autophagy genes, AT4, AT12, etc. So it's a pretty important linchpin for starting autophagy. And we know that autophagy is a really critical part of keeping our bodies functioning properly, getting rid of not just organelles and proteins that are misfolded and other things, but getting rid of even cells that are not functioning properly.
So it essentially, when you're under this stress, you're seeing that the ATP levels are a little bit down because we lack the nutrients around to be able to just go ATP, ADP back and forth pretty easily. We lack the energy, or the mitochondrial function is nutrient dense. When we lack that, when we go to a point where there's more AMP being produced and AMP kinase is more activated, that's going to turn on the FoxO3 nuclear transcription factor, phosphorylating it, telling it to go back into the nucleus, turn on these genes, and then we clean up and we go into scavenger mode. So AMP kinase creates its effect on the nucleus through the FoxO3 nuclear transcription factor, correct?
Exactly.
Perfect. So that's where the cycle activates. And then what happens is we scavenge, we clean up, we start to have maybe a little bit more raw material. Essentially, think of it like spring cleaning of your house, right? You're going around, you're gathering all the things that you can sell, you're gathering all the things that you wanted to toss, you're cleaning up your home and getting it ready for the next season. It just happens at a much higher rate within the cell. And I imagine there's certain circumstances that enable that to occur a little bit more readily. So let's talk about some practical situations where this kicks up a notch. Let's talk a little bit about exercise, fasting, and specific other potential factors that might drive increased AMP kinase, increased phosphorylation of FoxO3.
Yeah, obviously the things that are the sort of health tricks and biohacks that we've been talking about, things like exercise, although that probably shouldn't be a biohack. But things like, intermittent fasting for sure will activate it because you're activating AMP kinase. There are other ways to activate FoxO3 also, which may seem sort of counterintuitive, but I think if you understand it a little better, you'll see why. Inflammation from injuries or infections can also activate FoxO3. Now, it does it in a bit of a feedback control mechanism sense. So it's not directly causing it because, of course, inflammation is triggering and is triggered by upregulation of things like NF-kappa B, which is another nuclear transcription factor, but associated with generating lots of inflammatory cytokines and just the entire inflammasome. And it's not just infections or inflammation, also hypoxia. So if you are, if you were to drown or something like that, or spend some period of time with a low oxygen environment, you're going to trigger stress in the cell. Now, sometimes
this is the idea of like altitude training, for example, right? Where you train at a high altitude, which is a lower oxygen level, hypoxia, low oxygen, right? So a lot of athletes will go and train in high altitudes like Denver or Mexico City, and they'll train in those altitudes where there's a much higher altitude, and lower oxygen level. What that does is it trains the body through these particular genes to become more efficient and more effective at utilizing oxygen when it is available.
Exactly. The Kenyans are the greatest long distance runners, and they've got, I think an average altitude of 8,000 feet or something crazy that they're training in. There is another mechanism for activating FoxO3 also, which is interesting. It's DNA damage. So things like toxins or UV light or other radiation damage will cause a cascade of different responses in the cell to repair DNA. And it's amazing the amount of damage that DNA actually sustain every second of every day. I mean, there are literally thousands of point mutations and breaks and other things that are going on in the DNA. So really, there's always some level of DNA damage repair, or DDR, that is going on inside the cell, trying to maintain the integrity of our genetic code. And so, it's not 100% effective, but when that damage starts to increase, you see a promotion of FoxO3 to increase the expression of things like, GAD45. And what GAD is doing, and actually what GADD stands for, is growth arrest DNA damage repair. So what they're doing is these FoxO3 and the DDR genes that are being activated in GAD, what it does is it slows the cell's division down. And this is really, really important. So I don't want to gloss over it, because part of what FoxO3 promotes is a more quiescent state in stem cells and other cell types where you don't want to run through them. You don't want to use up all the potential of your stem cells and your progenitor cells. So let me walk through this really carefully. When you activate GAD, it's slowing down the cell reproduction process. It's arresting growth. And when that happens, there's more time for these DNA damage repair mechanisms to work, because it's sort of like you're running a race, okay? Or you're in a race car and you come in for a pit stop. You got to stop the car. You can't change the tires while the car's running. You got to stop it. And so that's what these guys are doing. They're stopping the growth in the cell. Now, when I said before, you don't want to use up the number of cycles that you can of stem cells, et cetera, what I'm talking about is something called the Hayflick limit, okay? Cells have a limit, a parent limit, to the number of times that they can divide. Mm-hmm. So if you've got a stem cell or a progenitor cell, it's supposed to be every time you need new cells or you need to replace damaged ones, or senescent cells are going to be killed off, you want to replace them. You've got stem cells that are going to provide that germ cell, if you will. It's not really a germ cell, but to produce the cell that you're looking for. It's producing that replacement cell. But it can only do that so many times based on what's called the Hayflick limit, because of a lot of different genetic issues, including telomere shortening, et cetera, that's going on. So you don't want to overdo that.
I almost think of that particular scenario as taking a photocopy of a photocopy of a photocopy, and slowly it starts to fade out, right? As you go too far down the line, you can't truly read what it says on that paper, on that third, fourth, fifth, sixth, seventh photocopy of the same image down the line, right? You still want to ideally have that original, but as you do it too much, it becomes problematic. So we don't want to go down that line so much. And so that's you're getting close to the limit. So when you slow it down, you remain utilizing the second or the third or the fourth copy in essence, or in an analogy that may be a little bit more understandable.
No, I like that analogy a lot. In fact, I think you can think about it in sort of two different directions. You start photocopying and you got plenty of ink in your cartridge, and it's just a bad lens or a bad printer head. And as a result, you take a clean copy and you make another copy. It's just as dark, but it's got more specs on it, and it's not, and the lines aren't perfect. And now you make a copy of that, and it's still just as dark because you had plenty of ink. And you slowly, it just becomes like static filled. Whereas the alternative is you don't have enough ink. And so when you make that first copy, it's a little bit lighter. And then the next copy is a little bit lighter. And before you know it, you're just spitting out white paper. There's nothing left on it. So those are two different ways that the system can fail just by having too many copies. I think of this a little bit more like the latter. So you're trying to make a copy, but you can't. It's not that it's a bad copy. It's just you literally can't make any more copies. And so what you want to do is minimize the number of copies that you have to make. And the way to do that is to activate this growth arrest or quieting down of the stem cells to preserve them, basically just to preserve them.
Yeah, making the current cells last longer, making them more efficient so that they don't need to be replicated as often. I think that's a great way to kind of look at it. I do want to just go back to, you mentioned inflammation, you mentioned fasting, exercise, all of these specific things. They're all, I believe, in general, hormetic stressors when done so in short periods and small amounts that essentially teaches the body to go into that activated state and then come out of that activated state, right? And that is key here. When you're talking about inflammation, you're not talking about chronic inflammatory reactions where you're constantly dealing with LPS coming in and stress response mode constantly going on and cell danger response turning on. And we're not dealing in a chronic scenario. We're very much dealing in these short bursts of small stress that trigger these initial stages of beneficial stress And then if we go too far, we go deep into that chronic stress state. What happens to the FoxO3 gene during those instances?
So that's really interesting and sort of is going to presage what we're going to talk about the autonomic nervous system and how it plays into the role. But you're absolutely right. When you have hormetic stresses, that's activating the sympathetic nervous system in short periods for healthy stress response. That does increase FoxO3 expression and activity. So yes, short periods of stress, going out for a run, go lifting, lift weights, go in a cold plunge, all those things will activate FoxO3 briefly. The problem is when it's chronic. It's that body's response to stress, which pushes you back away from, stress is knocking you out of homeostasis, and you're going to try to push back. Your sympathetic nervous system is going to try to push against that stress and push it back in the other direction, but it doesn't know when to stop. And so if you're chronically stressed, it's going to push you way to the other side and way out of homeostasis. Short periods, FoxO3 gets you back into homeostasis. When you have it going on for too long, it drifts, way out of commission, and FoxO3 levels actually get depleted. Mm-hmm. You actually see you run out of it. We'll talk about parasympathetic and how that's different in a minute, but I do want to spend a moment talking about the fine tuning of FoxO3 expression, because you got what we talked about here was FoxO3. Once it's there in the cell, you're moving into and out of the nucleus. When it's out of the nucleus, it's sort of quiet. It's there in reserve. And then when it's phosphorylated, serine 413 or other places, it will move back into the nucleus and begin doing its promotion of the stress response genes that we talked about. But what you also have is a fine tuning of how much FoxO3 there is within the cell in either place. And that's epigenetics. And so we've talked about the three most important ways that epigenetics play a role in controlling the amount of any given protein that's made. You got DNA methylation, and then you've got histone modifications, and then you've got microRNAs. And sure enough, all three of them play a role in managing this FoxO3 expression. So how much you have in your cell. With respect to DNA methylation, you want to have as little methylation as possible, because you want to be able to access that gene readily. So that's pretty easy. With the histones, there's lots of different ways you can modify histones. You can acetylate them, you can methylate them. There's other chemical modifications. What you want to do is you want acetyl groups put onto the histone 3. Okay, at lysine 18 and lysine 27. You want to have them maximally acetylated. There's a group of proteins called histone acetyltransferases. Name makes sense. It's working on a histone. It's putting an acetyl group onto it. It's an acetyltransferase. We call them HATs, HATs. They're really good for activating FoxO3. Now you have histone deacetylases. Those are the enzymes that take the acetyl group off. You don't want those. And then you have what are called histone methyltransferases. Okay, those are putting methyl groups on, and methyl groups tighten up the DNA around the histones. So whereas the acetyl group loosens the histone, makes the DNA easier to access, methyl groups make the histone, the whole nucleosome, tighten up and sort of lock the DNA away. So it's a bit of an oversimplification to say that this, HATs are good and histone deacetylases and HMTs, the methyltransferases, are bad. But it is true that if you have somehow impaired histone acetyltransferases, or you've activated the methyltransferases, you actually see a change in life expectancy and health span. So it's pretty amazing. And actually it's interesting because it also includes the gene, the BRCA1 gene. People always say, oh, I have the BRCA1 gene. Everybody has BRCA1. The question is whether or not you have a normal version of it or you have a mutated version. If you have a mutated version that's not functioning properly, well, that leads to hypermethylation because that's what BRCA1 does. It's involved in the methyl transferase. If you have hypermethylation, which is when you have that BRCA1 mutation, then you end up with this less FoxO3 available and you have a greater risk of cancers. So just an interesting little tie-in there.
Yeah, that's a great little tie-in to understand. So in general, and hyper-simplified, acetyl groups will loosen it and allow the DNA to be opened and for that FoxO3 gene to ideally be transcribed and for the protein to be created, versus methyl groups are going to tighten it up. So if we acetylate, we increase. If we methylate, we decrease the ability for that gene to be transcribed. And I believe that's true across the board for all genetics, period.
By and large, yeah.
By and large. This is epigenetics that's driving this. These are epigenetic factors that are driving these. Are there...
Yeah, this is really important also, just to take it one step further. We've talked about Yamanaka factors.
Yes.
Factors that actually help to rejuvenate cells, not just arrest the growth cycle so that you can live longer without needing to sort of run through your maximum amount of growth potential, but actually rejuvenating cells. There's groups like the David Sinclair's group up at Harvard that are trying to find small molecule drugs that will simulate that effect as well. And one of them that they're looking at is valproic acid. What is valproic acid? It modifies, among other things, it modifies histone side residue modifications, histone modifications. So it sort of ties it back. The more you modify that histone, the availability of that gene to be transcribed, the more you can potentially affect FoxO3 levels.
I think that's so huge to understand that FoxO3 is so important for being kind of that middle point of enabling so many important longevity-based factors to be managed and to slow down the replication cycles, to slow down almost the nuclear equivalent of the parasympathetic rest and digest kind of idea, right? Of
they go together.
Turning on that rest idea and putting your body into a state of self-recovery or enabling the recovery process to occur. And I think that might be a nice little lead into this topic of autonomic nervous system regulating or having some effect on the FoxO3 gene as well. Let's talk a little bit about how our autonomic state affects our genetic cycles and genetic capabilities to turn on that FoxO3 gene.
Sure. So when you're in a chronically sympathetically active state, so that chronic fight or flight state that we all want to avoid, that we've shown is pro-inflammatory and damaging, as I mentioned before, you get this short-term upregulation of FoxO3 because you're stressed, but you sort of burn it out. You burn it out and you see a decrease in FoxO3 expression, but you maintain that inflammation. And so that ends up causing an increase in reactive oxygen species production, mitochondrial dysfunction, damage to the epigenome. You end up with a higher level of methylated DNA, so you're locking stuff away. Or in some cases, you have exactly the opposite, which is suddenly the cell that's not supposed to make gene X, Y, and Z is now making, those proteins, and that can lead to cancers. But in the case of the parasympathetic nervous system, it's really a function of the fact that the parasympathetic is sort of quieting everything down. It's creating a state in which you don't need the FoxO3 to be expressed. You want to have it available, but you don't want it to be functioning because you don't want to be in stress. You don't want to be in that stress state. So it ties into cortisol as well. So cortisol is a big promoter of FoxO3 in the initial states, but over time, it causes, a burnout of FoxO3. It leads to apoptosis in the brain in models, of sort of chronic cortisol activity. You see like anywhere from a 30 to 50% decrease in hippocampal activity and connectivity. And it's actually, cortisol is, getting back to the epigenetics, cortisol is recruiting the histone deacetylases that we talked about. You want to have those histones acetylated so that you can have access to the gene or genes. So in general, inflammation is activated by this. And you actually see, mesenchymal, which you haven't talked yet about, which is really important, mesenchymal progenitor cells, sort of the stem cells that are involved in, basically everything associated with mesodermal cells. That includes fat, that includes bone. You see a shift when you have this chronic sympathetic activity. You see a shift away from muscle growth. You see a shift away from bone growth, and you see an increase in fat deposition and fat growth. So heightened sympathetic activity is actually wrecking your metabolism, which is one of the reasons why we say if you're in a chronically stressed state, it's almost like being, especially if you go without sleep, it's almost like becoming diabetic in a week because you're messing with your metabolism. And you actually see as you age and you have this sort of increase in inflammaging, you end up with people becoming more frail, especially in women. You see this, it's a big contributor to osteoporosis.
Totally. That makes a lot of sense. I like to think of the parasympathetic state as kind of the conserving and preparing state. We're generally a little bit slower. Our breath rate is a little slower. Our heart rate is a little bit slower. We're in this slowed down, conserving, holding on to, maintaining and preparing our cellular and our organ-based capabilities for when stressors do pop up. And we need to go into those stress states from time to time. And ideally in a flow and grow type of state, right? Ideally, we go into a sympathetic state where we're exerting or exuding these energies that we've conserved and prepared in the development or in the transaction towards a goal, right? We're trying to accomplish a goal. So cognitive function or exercise-based physical function that's driving us towards muscle growth or to bone or capabilities from a completion of a task or productivity standpoint. So we go into sympathetic state when we want to ideally perform this in an acute scenario, and then we can come back to that parasympathetic conserving state, ideally as often as possible. We don't want to slip off into the fight or flight state where it becomes chronically exerting energy, chronically burning through the conserved amount. And what that means is we go into a state where the FoxO3 becomes depleted. We go into a state where the inflammation is chronic, long-term, less than optimal, right? And that's where we want to avoid. If we go down that path, then ultimately the body can fall off into a state where it goes into complete just shutdown mode eventually. And that's kind of that dorsal vagal from the polyvagal theory. I'm doing the air quotes here. It's very much that complete shutdown state where we're not exuding any energy anymore. And that's where we really want to avoid. So
it's the end state of chronic sympathetic activation.
That's exactly right.
When
And
you just burnt out.
yeah, full burnout. So what we don't want to go down that path. What we want is short bursts of sympathetic activation followed by longer stretches of parasympathetic rest, digest, conserve activation. Because what that does is it balances out and it preps FoxO3 to be present and ready at good quantities for when the next stressor does come up. What we want is autonomic flexibility driving FoxO3 preparation and capability so that it can handle the next stressor to pop up. I think that is where we can put these two pieces together, because on the macro level, we're looking at the autonomic state, and on the micro level, when we go down to the genetic level, that's what we're trying to turn on, is that FoxO3 gene, and have enough of it available, have enough of the protein nuclear transcription factor available so that it can promote these cleanup crews and activation of autophagy and activation of these important factors of conserving and supporting and preparing when necessary.
I think that's exactly right. You want to have an ample supply of FoxO3, but you only want to have to use it for brief intervals where you've entered a stress state more as a hormetic stress, as opposed to a damaging stress that's prolonged. So yes, I think that's exactly right. Now, but I want to take this beyond, because it's great that parasympathetic activity will upregulate FoxO3 expression. It's great. It's awesome. It's one of the reasons why being in that parasympathetic recovery mode on a regular basis is actually geroprotective and helps promote longevity and a longer health span. And as you and I have talked about, there's a paper coming out that I'm one of the co-authors of all about this issue. But I want to take it one step further because it's been taken one step further and it's really, really exciting, which is that, over the last 3 years, there's been just a lot of discussion about how we can modulate FoxO3 activity. Because in aging, you lose FoxO3 through inflammaging and through epigenetic drifts and all sorts of other things and buildup of reactive oxygen species damage, some of which can be overcome with autophagy. But at the end of the day, there's just this inexorable drift to having less and less and less FoxO3 expression, which is why you see this sort of acceleration of aging-like symptoms as you enter your 7th and 8th decades. Well, over the past couple of years, and really there was an unbelievable paper that was published just this past September out of China, in which they had taken and through genetic manipulation, CRISPR tools, they had modified these mesenchymal progenitor cells, these stem cells associated with the mesodermal cells. And they injected or they upregulated FoxO3 production. So they artificially enhanced in those cells the production of FoxO3. And then they injected those cells back into some aged primates. So we're not even talking about mice or nematodes or anything like that. We're talking about primates, which, brings it really close to humans. And what they did a 44-week long study. So, I mean, that's a 9-month plus study looking at, okay, what's going on in these animals that have gotten this bolus of just one type of cell. I mean, yes, it's a stem cell, but a really important one. But it's still, how's that going to affect neurons? How's it going to affect, liver cells? How's it going to affect, your kidneys and your heart? And what they found was phenomenal. It literally reversed the aging in these aged animals. They took the equivalent of like retirees in the primate community, and they were able to reverse the age at the organ level, at the brain level, at the bone level, at the muscle level, even skin and fur coloring changed. Eyesight got better. All of these things, and not just a little bit. We're talking like 25 to 30% minimum across the board. Now, that's pretty, far along the translational, path to getting to the point where we could do this in humans. And the reason why that one cell type, beyond the fact that it can differentiate into healthier cells and promote stem cells lasting longer, these cells release what are called exosomes. And exosomes, for those of people who don't know, they're little bubbles, if you will, filled with whatever the cell wants to send out. Well, these mesenchymal stem cells are sending out these exosomes that are filled with geroprotective compounds, things like spermine and other things that act as senolytics. So they're killing off those zombie cells that have accumulated in the older body. So they're acting as senolytics, and in some cases even xenomorphics. So senolytics are compounds that will kill off senescent cells. They're literally lysing them, they're cutting them up. Xenomorphics will literally reverse the age of that cell just by introducing these exosomes and the contents into that cell. It sort of rehabilitates that cell, even though it wasn't, even though it wasn't specifically that cell wasn't created by this mesenchymal progenitor cell, but it's been affected by it through this release of exosomes. So just some really, really exciting work that's going on. I know there's Peter Attia has long talked about and really has promoted this idea that nothing is really going to extend human life beyond sort of the limit of 120 years. And I feel like that's probably been true. Yes, you can take things and do things that will enhance your health span, but the ultimate lifespan of a human seems to be limited around 120 years. But I believe that just within the last 5 to 10 years, we've seen two technology areas which are related to one another that actually have the ability not just to sort of keep us alive for as long as possible within those limits, but actually to break those limits. And we've talked about partial reprogramming a lot, and the Yamanaka factors and small molecule drugs and the partial reprogramming, some of the work that was done by OpenAI and Retro Biosciences to come up with new versions of the proteins that can do this. But I think this FoxO3 infusion of FoxO3 enhanced mesenchymal progenitor cells, which is easy to get because you can just filter your own out of your own blood. I mean, you can't do it, but I mean, labs can filter your blood, get those progenitor cells, modify them, and maybe we won't need to use CRISPR to do it, but maybe we can do it with chemicals to modify and upregulate FoxO3 and then just infuse those cells back into you.
Wow.
I mean, that would be pretty remarkable. And I think we're on the verge of being able to do that. In fact, I think they're planning human studies within the next year or so.
It's crazy to think how far we've come and where we're at now with the genetic capabilities and some of these amazing findings, and how quickly AI is helping to promote the understanding that we have to accelerate the findings and the capabilities that our healthcare system is going to be able to do, and our wellness care system. I think you're exactly right. We're breaking those limits that we initially had created or thought we had on us, right? That the ones that Peter Attia mentioned, the ones that we've kind of all attuned to. Living to 100 has been kind of the goal for the longest time. And now we're realizing that maybe 100 isn't the limit. Maybe 120 isn't even the limit. Maybe it's well beyond that. And maybe we are going to be far more capable of doing this. And seeing these studies where we're actually enhancing life, creating this sense of longevity, and almost reversing how much age has actually affected us or caused us to break down. We're really on the verge of something unique.
Well, I quote him often in my lectures on this. Ray Kurzweil wrote "Live long enough to live forever." That's really two pieces. You got to do all the right things so that you can remain healthy for as long as possible. That's the sort of thing that Peter Attia is promoting. But the other piece is to live forever. So you're absolutely right. You don't want to get to 100 and be sort of completely decrepit and broken down. You want to get to 100 and be strong enough so that you can rejuvenate yourself back to 20. I realize that people are going to say, oh, this is just hype and all that. And maybe it is right now. But I'm looking forward and saying, if we can imagine it, it will happen. And just the same way, if you go back to the 1920s and you look at those silent films where, people were shooting a ballistic, a bullet and riding a bullet to the moon, obviously that's crazy. Nobody would think you could do that. But yet, 60, 70 years later, we're in orbit, we're going to Mars, we've been on the moon, there's all of these crazy things. We're landing, rockets and on comets. So I think that if we can imagine it and we can see a path through things like partial reprogramming and FoxO3 enhancement, we can see a path to doing it. I believe that we will do it. So the trick is to live long enough so that at some point, maybe it's 15 years from now, when I'm, getting close to 70 years old or maybe a little past, I go in and I have a treatment that rejuvenates me back to being 25 or 30 again. And then I just progress normally through the next 50-year cycle and short of any traumatic, accident or something like that, there may not be a limit. There may not be a limit.
I know we're getting close to time, but I want to ask one more question. And I think it may be a lead to a whole new episode of this. Or talking about this, but has there been any talk about vagus nerve stimulation specifically with regards to the FoxO3 gene? Has there been any link between VNS and FoxO3 activation?
Yes, there has, and that's it's in in my lecture, and it's going to be in my in the paper. Yes, vagus nerve stimulation, just like any other parasympathetic recovery technique, will increase the production of FoxO3, increase and enhance FoxO3 expression. So it's one of the reasons why I believe that vagus nerve stimulation is very much a tool for those who are looking to biohack to a longer, healthier health span, I'm not saying it's going to do the age reversal, but there are some signs that at least some of the hallmarks of aging do reverse when you're in that parasympathetic recovery mode triggered by vagus nerve stimulation.
I think that's huge. And when the paper comes out, I'd love for us to sit down again. I know we've released the episode on autonomic deregulation theory of aging, which is what your book is on, but I'm excited, the paper, excuse me, but I'm very excited to, the book is coming, I imagine. Yes. And I would love to dig deeper into this, because now that we can understand these mechanisms, I think we can truly take this to the next level and really understand and create systems around longevity that involve autonomic tools, that involve mitochondrial support tools, and that involve potentially some wonderful tools that can enhance genetic capabilities through tools like FoxO3. So
agreed.
I think we're on the verge of something, and I'm excited to be a part of it with you.
I am too. This is an exciting time to be alive. We are approaching the singularity from many, many different directions. It's pretty exciting.
Absolutely. I think this has been a really cool episode. For those who are interested on the genetic side of things, dig into FoxO3, dig into the tools like vagus nerve stimulation that we've discussed. Check out some of the past episodes, because I think they'll all heavily relate to this particular topic. And if you got to this point in the podcast and you're interested in learning more, you can share this with one person who you think could utilize this information, and dig into past episodes, because there's a lot of great information that we've talked about over the past 130 some odd episodes that we've done that may be of very high interest and high value to all of you. Thank you so much for listening. We'll catch you all on the next one.